Today, we are tackling something that I think most of us, well, we just take it for granted. We treat it like a background character in the movie of our lives, maybe a bit of an extra that we only notice when things go wrong.
That's so true.
But it turns out it's actually the director, the producer, the craft services team, and the janitorial staff all rolled into one. We are looking at the liver.
It is arguably the most underappreciated organ in the entire human body. I mean, we tend to think of the heart or the brain as the superstars, the flashy ones. But the liver is this massive, silent industrial complex that makes everything else possible.
And we aren't just skimming the surface today. We're doing a proper deep dive decoding Chapter 71 of our medical text. We're talking about the largest solid organ in the body. I knew it was big, but the source material says 1.5 kilograms.
Which is about 3.3 pounds.
3.3 pounds. And the average adult, that's significant.
It really is. Imagine carrying around, I don't know, a brick or maybe a very small chihuahua inside your abdomen. That's the weight we were talking about.
A chihuahua is a great visual. But unlike a chihuahua, this thing is doing actual work.
A lot of work. And the mission for this deep dive is to understand why this organ is so heavy, both metaphorically and literally. It isn't just a filter. I think that's the common misconception, that it's just a strainer for booze.
Right. That's what everyone thinks.
But it's a chemical factory. It handles digestion, metabolism, immunity, and storage all at the same time, in the same building.
That's the hook for me.
Yeah.
That it performs functions that seem completely unrelated to each other. It's cleaning your blood while simultaneously baking energy cakes for your muscles and storing vitamins for a rainy day. So set the scene for us. What happens if this factory shuts down?
If the liver fails, the system crashes. I mean, immediate chaos. We're talking about everything from your blood losing the ability to clot.
So you just bleed out.
You bleed out. To your brain being poisoned by ammonia, to your energy levels just completely flatlining because you can't process sugar. The liver is a discrete organ, physically, but functionally. It's woven into every single system. If it goes offline, you don't just get sick. You stop functioning as a biological entity.
Okay, that definitely raises the stakes.
Mm-hmm.
So let's unpack this thing, and we need to start with the visuals. If I were to shrink down, magic school bus style, and walk inside the liver, what am I looking at? Because the source material describes a very specific structure called the liver lobule. It sounds like the basic Lego brick of the organ.
Right. This is the basic functional unit. And you're right. To understand the liver, you have to stop thinking of it as just this slab of meat. You have to imagine it as a modular city. A human liver is made up of anywhere from 50,000 to 100,000 of these individual districts called lobules.
50 to 100,000, that's a lot of districts. And they are small, right? Really small.
Correct. They are microscopic processing plants, just a few millimeters in length and maybe 0.8 to 2 millimeters in diameter.
Okay, so I'm standing in this microscopic city. What's the layout? Is it a grid, like Manhattan?
No, it's circular. The best way to visualize a lobule is to think of a wheel, like an old-fashioned wagon wheel or maybe a bicycle wheel. That's the core concept.
Wheel? Okay, I can picture that. Let's break that analogy down. Yeah. I'm standing at the very center, at the hub. What is the hub?
The hub of that wheel is the central vein. Everything in the lobule is organized around this central drain. It is the exit point for all the processed blood.
Okay, so I'm standing on the drain, I'm looking out from the center, and I see spokes, right?
Exactly. The spokes are the hepatic cellular plates. These are basically walls made of the liver's main working cells, the hepatocytes, and they radiate outward from where you are standing at the hub, going all the way to the rim. These walls are usually about 2 cells thick.
So you've got these living walls radiating out. What's on the rim of the wheel? The outer edge.
The rim is what we call the interlobular septa. This is the fibrous connective tissue that separates one lobule from its neighbor. It's kind of like the property line between districts. And inside this rim, dotted around the perimeter, you find the portal triads.
The portal triads? That sounds like some kind of sci-fi organization.
It does, doesn't it? But it's really just the plumbing intake. Triad means 3. So in these septa, you have 3 distinct tubes bundled together. A small portal venule, a hepatic arteriole, and a bile duct. This is where all the supplies arrive for processing.
Okay, so let's talk about the flow. In a factory, you have raw materials coming in and products going out. How does fluid move through this wheel structure?
This is absolutely critical to understanding how the liver works. The flow of blood is centripetal. It moves from the rim of the wheel toward the hub,
From the outside in.
from the outside in. Blood comes in from those portal venules, which are carrying blood directly from your gastrointestinal tract, and from the hepatic arterioles, which are carrying fresh oxygenated blood from your heart.
Wait, hold on a second. You've got the vein from the gut and the artery from the heart coming in at the same spot,
Yes.
and they mix,
Yes.
because that seems unusual. Usually the body tries very hard to keep oxygenated arterial blood separate from the deoxygenated, used-up stuff from the veins.
That's the magic of the liver. They mix. You see, the liver is greedy. It needs the oxygen from the artery to fuel its own incredible machinery. But it needs the raw materials, the nutrients, the toxins, the chemicals that are coming in from the gut vein. So right there at the rim of the wheel, these two completely different streams of blood merge together.
So where do they go once they've mixed?
They flow into these flat branching channels called hepatic sinusoids. These sinusoids lie between those cellular plates, the spokes we mentioned. Think of them like the canals in Venice, winding their way between the rows of houses.
Ah, okay. So the blood is just washing over the liver cells like a tide coming in, moving continuously from the outer rim toward that central vein where I'm standing.
Precisely. The liver cells are just bathing in this mixed blood. They are constantly exchanging fluids and molecules with the blood in the sinusoids. They're grabbing what they need, like glucose and amino acids, and they're dumping what they don't, like urea and other waste products. And then once the blood has been processed, it drains into the central vein, which eventually feeds into the hepatic veins and then back to the vena cava to go to the heart.
It sounds incredibly efficient. But there's a second flow happening, right?
Yeah.
You mentioned a bile duct in that triad on the rim.
Yes, and this is where the traffic gets really interesting. Bile flows in the exact opposite direction. It's centrifugal. It moves from the center outward.
So it's a two-way street, a countercurrent system.
Exactly. The liver cells secrete bile into these tiny, tiny spaces between them called bile canaliculi, and that bile then flows outward, away from the central vein, to the bile ducts in the rim. So you have this constant opposing traffic, blood rushing in toward the center, and bile slowly moving out toward the edge.
That is a remarkably complex and elegant design for just a few millimeters of tissue. It's beautiful, really.
It is. And that efficiency relies heavily on the plumbing system, which brings us to the vascular physiology. I mean, the numbers here are honestly staggering when you start to think about them.
Okay, let's look at those numbers. The source says the liver receives 1,350 milliliters of blood per minute.
That is massive. To put that in context for you, that is 27% of your resting cardiac output.
Almost a third. A quarter of all the blood your heart pumps with every beat is going straight to the liver.
More than a quarter of every single heartbeat is detoured to the liver for processing. It is a high-volume, high-priority system.
And it's split. You mentioned most of it isn't coming from the heart directly.
Right. About 1,050 milliliters of that comes from the portal vein. That's the venous blood from your stomach and intestines, packed with everything you just absorbed. Only about 300 milliliters is fresh arterial blood from the hepatic artery. But here is the key engineering marvel of this whole system: the pressure.
Okay, let's unpack the pressure. Because usually when you shove that much fluid through a small, complex filter, pressure goes way up. It's like putting your thumb over the end of a garden hose.
Exactly. Resistance creates pressure. But the liver has unbelievably low resistance. The pressure in the portal vein as it's entering the liver is only about 9 millimeters of mercury.
Which is really low for a vein, right?
It's very low. But the pressure leaving the liver in the hepatic vein is about 0 millimeters of mercury.
0.
Effectively 0. So the pressure drop across this massive, complex organ is tiny. A huge volume of blood over a liter every single minute glides through this organ with almost no friction. The sinusoids are wide and accommodating. It's less like a filter and more like a slip-and slide.
Until it isn't. Because we have to talk about what happens when this plumbing gets clogged. The source brings up cirrhosis. This is where the story takes a dark turn.
Yes. Cirrhosis is essentially the destruction of that beautiful architecture we just described. The liver cells, the parenchymal cells, they get destroyed and are replaced by fibrous tissue.
Scar tissue, basically.
Exactly. And scar tissue acts like concrete. Imagine taking that soft, spongy, low-resistance slip and slide and pouring quick, dry cement all over it.
And the scar tissue actually contracts, doesn't it? It shrinks and tightens.
It contracts and tightens and strangles the blood vessels. Suddenly, that low-resistance slide becomes a high-resistance obstacle course. It becomes a dam.
And what causes this? I mean, we all know alcohol is a big one.
Alcoholism is a major cause for sure, but also poisoning by things like carbon tetrachloride or viral hepatitis, or even a long-term bile duct obstruction. But what we are seeing a massive rise in now is NASH, non-alcoholic steatohepatitis, and its precursor, NAFLD, non-alcoholic fatty liver disease.
Which is associated with obesity and type 2 diabetes.
Correct. It is actually the most common cause of liver disease in the United States right now. Excess fat accumulation in the liver leads to inflammation, which over time leads to fibrosis. And when that resistance rises, no matter the cause, you get portal hypertension.
The blood simply can't get through.
It can't get through. It hits a wall. The pressure in that portal system, which should be a gentle 9 mm Hg, jumps to 15, 20, or even higher. And that leads to a cascade of terrible, life-threatening effects.
Before we get to the really bad stuff, there's another cool function of this vascular system that acts as a kind of safety valve. The liver acts as a blood reservoir.
It really does. It acts like a sponge. Normally, it holds about 450 milliliters of blood, which is roughly 10% of your total blood volume. It's just sitting there kind of hanging out in the sinusoids. But if the pressure in the right atrium of your heart goes up, say in congestive heart failure, the liver can expand.
So it takes one for the team, essentially.
It does. It can swell up and hold an extra half a liter to a full liter of blood. It acts as this incredible buffer for the heart, storing that excess volume so it doesn't back up and overwhelm the circulation and cause congestion in the lungs or elsewhere.
That is wild.
Yeah.
It's physically getting bigger to protect the heart. Now, you mentioned pressure rising earlier. This connects to what the source calls the sweating function of the liver. I, for one, did not know the liver sweated.
Sweating is a very vivid way to put it, but physically, it's remarkably accurate. To understand this, we need to talk about lymph formation. The pores in the walls of the hepatic sinusoids, those channels where the blood flows, are huge.
How huge are we talking?
About 1 micrometer. In the world of cellular biology, that is a gaping hole. It's like leaving the barn door wide open. It means fluid and even large proteins can diffuse easily out of the blood and into something called the space of Dissy.
The space of Dissy. That sounds like another sci-fi location.
It's the narrow parasinusoidal space between the blood vessel wall and the liver cell itself. It's like a little moat around the cells. And because those pores are so big, the lymph that drains from the liver has a very high protein concentration, about 6 grams per deciliter. That's almost the same as blood plasma itself.
And the liver produces a lot of this lymph.
It produces half of all the lymph formed in your entire body under resting conditions. Half. From this one organ.
Wow. Okay, so what happens when the pressure goes up, like in that cirrhosis scenario we were just talking about?
The liver starts sweating profusely. If the hepatic vein pressure rises just 3 to 7 millimeters of mercury above normal, the liver starts dumping excessive fluid into the lymph. If the pressure hits 10 to 15 millimeter Hg, lymph flow increases 20-fold.
20 times the normal amount.
Yeah.
Where does it all go? The lymphatic vessels can't handle that kind of volume.
They absolutely can't. They get completely overwhelmed. So the fluid has to go somewhere. It literally leaks or sweats through the outer capsule of the liver and drips directly into the abdominal cavity. That is ascites.
So that fluid-filled, distended belly you see in patients with severe liver disease, that's essentially plasma sweating right off the surface of their liver.
It is almost pure plasma, 80 to 90% protein. It's a tragic mechanical failure of the plumbing system caused by high pressure. Imagine a sponge being squeezed so hard it just weeps fluid continuously into the space around it.
That is a very sobering image. It really highlights how mechanical this biology is. It's not magic, it's just pressure and pipes and blockages.
Exactly. It's physics.
Okay, let's pivot from physics to something a bit more action-packed. Section 3 in our source. The security team. We established that all that blood from the gut goes straight to the liver.
Yeah.
But gut blood is dirty blood, right? It's full of bacteria.
It is incredibly dirty. We don't like to think about it, but our colon is full of bacteria. A sample of blood taken from the portal vein before it hits the liver is often teeming with colon bacilli.
Teeming, as in you can see them under a microscope swimming around.
Teeming. If that blood went straight into your general circulation, straight to your heart and your brain, you would be septic constantly. You'd be sick all the time. Your immune system would be completely overwhelmed.
So who is the bouncer at the door?
The Kupffer cells. They are also known as reticuloendothelial cells. These are large, specialized macrophages that are anchored to and line the hepatic sinusoids.
So they are sitting right there in the bloodstream, just waiting.
They are waiting. They are anchored to the walls of the sinusoids, and they are incredibly efficient phagocytes, which is a fancy way of saying they eat bacteria.
The source mentions high-speed motion pictures of this process. That must be something to see.
Yes, they filmed this. When a bacterium comes into contact with a Kupffer cell, the cell extends a pseudopod, grabs it, and digests it in less than 0.01 seconds.
Less than 1/100th of a second.
Yeah.
That's instant.
It's faster than a blink. It's practically instantaneous. The efficiency is just terrifyingly good. The source material states that less than 1% of the bacteria entering from the gut typically survive the passage through the liver to reach the systemic circulation.
So it's a kill zone, a microbial gauntlet.
It is a near-perfect filter. Essentially, the liver is the firewall between the filth of the digestion process and the sanctity of your systemic circulation.
That's amazing. Without Kupffer cells, we wouldn't survive a single meal. We'd eat a sandwich and die of sepsis an hour later.
That's not an exaggeration at all. It's a critical, non-stop security detail.
Okay, that is amazing. Moving on to section 4. This is the stuff that sounds like science fiction or Greek mythology. The regeneration superpower. We're talking about Prometheus, right?
Yeah. Right. The story of Prometheus, who steals fire from the gods. As punishment, Zeus chains him to a rock, and every day an eagle comes and eats his liver. And every night the liver grows back so the eagle can come and eat it again the next day.
Horrifically gruesome, but accurate.
Surprisingly accurate. The ancient Greeks were definitely onto something. The liver possesses a remarkable, almost unique ability to restore itself. The source says you can surgically remove up to 70% of the liver and it will grow back to its original size.
70%. That implies you could donate more than half your liver to someone and be fine.
You can. That's the entire basis of living donor liver transplants. In rats, this restoration process takes only 5 to 7 days. In humans, it takes a bit longer, obviously, but the mechanism is fascinating. It doesn't just grow back the piece you cut off, like a lizard's tail.
It doesn't. It doesn't just sprout a new lobe.
No. The remaining lobes undergo compensatory hypertrophy. They enlarge. They get bigger to make up for the lost mass. It's volume restoration, not shape restoration. The liver just grows and grows until it hits the right mass for your body.
So what triggers this? How does the liver know, hey, I'm missing a chunk, time to grow?
It's a complex dance of chemical signals. One of the main ones is called HGF hepatocyte growth factor. It's produced by mesenchymal cells in the liver and other tissues. After a partial hepatectomy, levels of HGF in the blood can rise 20-fold.
So the system basically screams, grow now, at the remaining liver cells.
Exactly. It initiates cell division on a massive scale. There are other players too. Epidermal growth factor, cytokines like tumor necrosis factor and interleukin-6, they all jumpstart the replication process.
But just as important as starting is stopping. How does it know when it's big enough? You don't want a liver the size of a watermelon filling up your whole abdomen.
That's the mystery of the liver to body weight ratio. The body has a set point. It knows exactly how much liver it needs for your specific body size. And once the liver reaches that correct size, a cytokine called transforming growth factor beta, or TGF-beta, acts as the powerful inhibitor.
It slams on the brakes.
It slams on the brakes hard. It completely shuts down the growth process. It's a perfectly tuned feedback looped system.
Is there a catch? It sounds too good to be true.
There's always a catch. Regeneration fails if the remaining liver is diseased. If there's viral inflammation or fibrosis, like in cirrhosis, the regeneration signal gets scrambled or the cells just can't respond. It only works well on healthy tissue.
Got it. So you can't count on regeneration if the liver's already fighting a war on another front. Now let's get into the meat of our deep dive. Section 5. The chemical factory. Metabolism. This is where the complexity really ramps up.
This is why the liver is the metabolic capital of the body. The cells here are biochemical wizards. They share substrates and energy between metabolic systems. It's not just doing one thing, it's juggling carbs, fats, and proteins all at once. Let's start with carbohydrates. The liver acts as a critical buffer.
The glucose buffer function. Explain that to me.
Okay. You eat a meal. Let's say you have a donut and a coffee. Your blood glucose spikes. High blood sugar is actually quite dangerous. It's toxic, it damages blood vessels and nerves over time. So the liver steps in, it grabs that excess glucose out of the blood and polymerizes it, converting it into glycogen for storage.
It's basically putting money in the bank for a rainy day.
Exactly. It stores it away. But then a few hours later, between meals, your blood sugar starts to drop. The liver senses this, depolymerizes that glycogen back into glucose, and releases it into the blood to keep your level stable.
So it keeps the lights on when the power starts to go out.
Yes. It maintains a steady state.
Right.
But it goes even deeper. If you run out of glycogen, say you're fasting for a long time or doing intense exercise, the liver performs gluconeogenesis.
Which means
It means new glucose creation. It literally takes other molecules like amino acids from proteins and glycerol from fats, and through a series of chemical reactions, it turns them into brand new glucose. It can even convert other sugars like galactose and fructose into glucose. It will do whatever it takes to maintain that blood sugar level.
And why is this so critical? Why not just let the sugar drop?
Because your brain runs almost exclusively on glucose. Without this buffering function, the source says your blood glucose would spike 3 times higher after a meal, which is incredibly dangerous, and then crash to fatal levels entirely between meals. You would literally go into a coma and die just from skipping lunch.
So the liver is basically the climate control system for our body's energy levels. It smooths out all the peaks and valleys.
That's a perfect way to put it. Now let's talk about fat metabolism. This is the big energy source.
I always associate fat with storage, not necessarily immediate energy.
And that's true, but the liver is the master of beta oxidation. It takes fatty acids and splits them into 2 carbon units of acetyl-CoA. This acetyl-CoA then enters the citric acid cycle to produce tremendous amounts of ATP, or energy.
More energy than the liver itself needs.
Way more. The liver performs beta oxidation much faster than most other tissues. It doesn't just make energy for itself, it exports it. It turns any excess acetyl-CoA into acetylacetic acid. This acid is soluble, so it travels in the blood to other cells, like muscle cells, which can then convert it back into acetyl-CoA and use it for fuel.
It's like an oil refinery sending out tanker trucks full of gasoline to all the gas stations in the country.
A perfect analogy. And it also synthesizes fats. It makes about 80% of the cholesterol in your body and also phospholipids.
We usually think of cholesterol as the bad guy.
But it's absolutely essential for cell membranes. We would literally fall apart without it. The liver makes it, converts most of that cholesterol into bile salts, which we need for digesting fats, and then ships the rest out in lipoproteins to help build and repair cells all throughout the body.
And what happens if we eat too many carbs? Like way too many donuts?
The liver is the culprit there too. It synthesizes fat from excess carbohydrates and proteins. It converts that sugar into triglycerides, then transports that fat to your adipose tissue to be stored as body fat.
So the liver is the one packing my suitcase when I overeat. Thanks a lot, liver.
It's just trying to save energy for later. But the most critical, the most immediately life or death metabolic function, the one that will kill you fastest if it fails, is protein metabolism.
The source is very clear on this. The body dies in days without this function, not weeks, days.
Yes, this is non-negotiable. First, deamination. To use amino acids for energy or to turn them into fat or glucose, you have to remove the amino group, the nitrogen part. The liver is the primary site for this.
And that deamination creates a toxic byproduct, right?
It creates ammonia, and ammonia is highly, highly toxic to the central nervous system. It's the same stuff in a floor cleaner.
This brings us to urea formation, which sounds incredibly important.
This is the lifesaver. The liver takes that toxic ammonia plus ammonia that's produced by bacteria in your gut, and it combines it with carbon dioxide to form urea. Urea is much safer, it's chemically stable, it dissolves in the blood, goes to the kidneys, and is safely peed out.
So the liver effectively puts a chemical straitjacket on the ammonia.
Exactly, it neutralizes it. But if the liver stops doing this, in end-stage liver disease,
the straitjacket comes off.
and the ammonia builds up in the blood, it crosses the blood-brain barrier like it's not even there. You get confusion, lethargy, and eventually hepatic coma and death. This is often how end-stage liver failure patients die. The ammonia essentially shuts down the brain.
That is terrifying. It really highlights how we are walking this fine tightrope of chemical balance every single day. What else in the world of proteins?
Plasma proteins. The liver forms 90% of all the proteins floating in your blood plasma. The only major ones it doesn't make are the gamma globulins, the antibodies, which are made by your immune system's plasma cells.
90%. That includes albumin, right? Which is the protein that helps keep fluid inside our blood vessels.
Yes. Albumin acts like a molecular sponge in the blood, holding onto water via oncotic pressure so it doesn't leak out into the tissues. If the liver fails and stops making albumin, plasma protein levels drop, fluid leaks out of the blood, and you get massive swelling, or edema.
But the amazing thing is, if you lose a bunch of plasma protein, say, from a severe bleed, the liver can replenish them in just a week or two.
It ramps up production like crazy. The hepatocytes undergo mitosis to expand their capacity and just start cranking out proteins. It really is a factory. It never, ever stops.
And it creates non-essential amino acids, too, on demand.
It can synthesize them from scratch using a process called transamination. It's chemically just incredibly versatile.
Okay, let's move to Section 6, the storage locker and the pharmacy. We touched on glycogen storage, but it stores a lot of other things, too.
It's a vitamin warehouse. It has a particular propensity for storing fat-soluble vitamins. It stores vitamin A in huge quantities.
How huge are we talking?
Enough for up to 10 months. You could not eat a single speck of vitamin A for nearly a year, and your liver would keep you covered.
Wow. Why would we evolve a system with that much backup?
Think about our ancestors. They didn't have a grocery store with carrots year-round. They might go for months without finding a rich source of vitamin A. The liver allowed them to survive those lean times.
That makes perfect evolutionary sense. What else does it have in the pantry?
Vitamin D for 3 to 4 months, and vitamin B12 for at least a year, maybe several years.
That explains why deficiencies in those vitamins can take such a long time to show up. The liver is buffering us from our own bad diet.
Exactly. It also stores iron. Except for the iron that's in the hemoglobin of your blood, the greatest proportion of iron in the body is stored in the liver in the form of ferritin.
How does that storage system work?
Liver cells contain a protein called apoferritin. When there's excess iron available in the blood, it binds to apoferritin to form ferritin.
So it's like a magnetic trap for iron atoms.
In a way, it acts as a blood iron buffer. If iron levels in the blood drop, the ferritin releases the iron back out. This is why liver, as a food, is such a nutrient-dense thing to eat. It's full of all these vital stores.
Makes sense. Now, coagulation factors. This is the stopping the bleed part of the job.
The liver produces almost all of the clotting factors. We're talking fibrinogen, prothrombin, accelerator globulin, factor 7, the list goes on and on. But there's a key ingredient that the factory needs to make these products.
Vitamin K.
Vitamin K. The liver needs vitamin K to manufacture these factors. If you don't have vitamin K, or if the liver is too damaged to use it, coagulation fails. You bleed uncontrollably from minor cuts.
This is why doctors check the prothrombin time, or the INR, in liver patients,
Right.
to see if the factory is still making the glue that makes blood clot.
Exactly. It's a direct measure of the liver's synthetic function. If that time is prolonged, it means the factory is failing to produce.
And finally, in this section, detoxification, the pharmacy and waste disposal unit.
The liver is the body's primary way of getting rid of drugs, toxins, and excess hormones. It detoxifies things like sulfonamides, penicillin, erythromycin.
So when you take a pill, the liver is what determines how much of it actually stays in your system and for how long?
Yes. It's called the first pass effect. For many oral drugs, the liver breaks down a huge chunk of the medication before it even reaches the rest of the body, and it also alters your own hormones: thyroxine, estrogen, cortisol, aldosterone. The liver breaks them down or chemically alters them so they can be excreted, usually into the bile.
And what happens if the liver is damaged and can't do this?
The hormones accumulate. You get an overdose. For example, if a man has severe liver failure, he might start developing feminine characteristics like gynecomastia because his liver isn't breaking down the small amounts of estrogen all men produce. Or you might get massive fluid retention because aldosterone, which tells your kidneys to hold on to salt and water, isn't being cleared.
So it's basically an overdose of your own natural hormones because the drain is clogged.
Precisely. It leads to overactivity of these powerful hormonal systems.
Okay, we are arriving at our final major section. Section 7, the diagnostic detective. We're going to talk about bilirubin and jaundice. This is usually the most visible, the most dramatic sign of liver trouble when a person turns yellow.
Jaundice, it's a classic, unmistakable sign. But to understand why you turn yellow, we have to trace the life and death of a red blood cell.
Okay, a red blood cell lives for about 120 days. Then it gets old, fragile, and it bursts. What happens next?
When it ruptures, it releases all its hemoglobin. Macrophages, the body's cleanup crew, found in the spleen and liver gobble up that hemoglobin and split it into two parts: heme and globin.
Okay, globin is just protein, so that gets broken down into amino acids and recycled. What about the heme part? The iron-containing part.
The heme ring is opened up. It releases its free iron, which then gets transported by transfer and back to the bone marrow to make new red cells, or to the liver for that ferritin storage we talked about. What's left behind is a straight chain of 4 pyrrole nuclei.
And this chain is the pigment.
Yes. First, it's converted into biliverdin, which is actually green. Then it's quickly reduced to bilirubin, which is that familiar yellow-orange color.
So this is the birth of bilirubin.
Mm.
Now it has to take a journey through the body.
And this journey is absolutely crucial for understanding diagnostics. This newly formed bilirubin is called unconjugated or free bilirubin. It's released into the plasma, but here is the catch. It's not water-soluble. It can't dissolve in blood on its own.
So it needs a taxi to get around.
It needs a taxi. It immediately binds tightly to plasma albumin, and because it's bound to this big protein, it cannot be excreted by the kidneys. It's just too big to fit through the kidney's filter.
So it has to go to the liver to be processed.
It has no choice. It goes to the liver. Inside the liver cells, it detaches from the albumin. The liver then performs a chemical reaction called conjugation. It usually attaches a molecule of glucuronic acid, about 80% of the time, or sulfate, 10%.
And conjugated basically means packaged for export, right? It makes it water soluble.
Yes, that is the key change. Now that it's water soluble, the liver can actively excrete this conjugated bilirubin into the bile, which then flows into the gut.
And once it's in the gut.
The bacteria in your intestines get to work on it. They convert it into a colorless substance called urobilinogen.
And this is where the pigment's path splits.
Right. Some of that urobilinogen is oxidized by gut bacteria into stercobilin. This is what gives feces its characteristic brown color.
So brown poop is actually a sign of a healthy, functioning liver and bile duct system.
It means your bile ducts are open and pigment is getting through. Exactly. It's a good sign.
What about the other path for urobilinogen?
Some of that urobilinogen is reabsorbed from the gut back into the blood. Most of that goes back to the liver, but a small fraction goes to the kidneys, where it is oxidized to form urobilinin. This is the pigment that gives urine its yellow color.
Yellow urine, brown poop, all thanks to the final breakdown products of old red blood cells. Now let's talk about jaundice. This is what happens when bilirubin gets stuck and builds up in the extracellular fluid.
If bilirubin levels in the blood rise above about 1.5 milligBL, which is roughly 3 times the normal level, it starts to deposit in the tissues, and the skin and the whites of the eyes turn yellow. We can classify jaundice into 2 main types based on what we just discussed: hemolytic and obstructive. The Vandenberg test helps us tell them apart by measuring the 2 types of bilirubin.
Let's break them down. Type 1, hemolytic jaundice.
Hemolysis means destruction of blood cells. In this case, your red blood cells are dying way, way too fast. Maybe it's transfusion reaction, maybe a disease like malaria or sickle cell anemia.
So the liver factory is working just fine, but it's completely swamped with raw materials.
It's overwhelmed. It can't conjugate all that bilirubin fast enough. So the levels of unconjugated or free bilirubin in the blood skyrocket.
And because the liver is still working on overdrive, it's also pumping out a lot of conjugated bile too, right?
Yes. It's working as hard as it can. So you have a very high rate of urobilinogen formation in the gut. This means you'll have very dark feces because of all that extra stercobilin pigment, and you'll also find a lot of urobilinogen in the urine.
Okay. Now contrast that with type 2, obstructive jaundice.
This is a plumbing blockage. A gallstone, or more ominously, a tumor, blocks the common bile duct. The drain is plugged.
So the liver does its job perfectly. It takes up the bilirubin, it conjugates it, but the finished product has nowhere to go. It can't leave the factory.
It hits a wall. The conjugated bilirubin builds up inside the liver cells until they actually rupture, and it leaks back into the blood and the lymph.
So now the blood tests are going to show very high levels of conjugated bilirubin.
Correct. And here is the classic clinical tell. The clay-colored stool.
Clay-colored.
Gray, white, pale. Because the bilirubin is completely blocked from reaching the gut. No bilirubin in the gut means no stercobilin is formed. The stool loses all its color and looks like gray clay. It's a very distinct, very alarming sign.
And what does the urine look like in this case?
Remember, conjugated bilirubin is water-soluble. Even though it can't get into the gut, it's floating around in the blood. The kidneys can filter this type, so the urine becomes intensely yellow or even dark brown. It can be foamy.
But the urobilinogen level in the urine would be 0.
Exactly, because no bilirubin ever got to the gut to be converted into urobilinogen in the first place. This specific combination, high conjugated bilirubin in the blood, clay-colored stool, dark urine, and 0 urobilinogen in the urine, is the unmistakable signature of obstructive jaundice.
That is a brilliant piece of diagnostic detective work. Just by looking at the color of a person's stool and urine, and checking which type of bilirubin is high in their blood, you can tell if the problem is with the blood cells being destroyed too fast or a physical blockage in the plumbing.
It's physiology in action. It allows us to pinpoint the problem without even having to cut the patient open. It's a beautiful example of how understanding the system lets you diagnose its failures.
So we've toured the factory. We've seen the architecture, the plumbing, the security team, the power plant, the warehouse, and the waste management system.
It really is the ultimate multitasker, isn't it? There's no other organ like it.
What stands out to me, wrapping this all up, is the fragility of the balance. We talked about pressure gradients of just a few millimeters of mercury. If that shifts by a tiny amount, your abdomen fills with fluid. We talked about ammonia conversion. If that one chemical reaction stops, you enter a coma.
And that's the takeaway, I think. The liver handles the most toxic substances, ammonia, drugs, bacteria, so the rest of the body doesn't have to. It takes the hit for the team, it absorbs the fluctuating blood volume to save the heart, it prevents the sugar crash to save the brain.
It's the silent guardian, working tirelessly in the dark.
It is, and consider this as a final thought. A little bit of scar tissue, a bit of fibrosis, can change the fundamental physics of the whole human being. It changes the pressure, the fluid dynamics, the chemistry, the hormones. It's a powerful reminder that we aren't just a collection of discrete parts. We are a dynamic flow of fluids and chemicals. And the liver is the master regulator of that flow.
Well, I will certainly never look at my liver, or for that matter, a glass of wine, the same way again. It's working incredibly hard down there.
It certainly is, every second of every day.
Thank you for guiding us through this amazing factory. And thank you to the listener for diving deep with us. Hopefully you've got a new appreciation for the chemical wizardry happening under your ribcage right now.
Always a pleasure.
Until next time, stay curious.